Hello everyone, and welcome to our lecture on microscopy using microscopes and some cell morphology. I'm going to break this lecture up into two separate videos just so we don't get too much video fatigue. In the first half we will cover microscopy, and we cover this in more detail than most other courses simply because it is microbiology and we have to use microscopes in order to see the organisms. So we're going to talk about the different types of microscopy and a few characteristics of each so that you will be able to select the correct microscopic tool to see what it is that you need to see in microbiology. In the second half of this lecture, we will cover cell morphology, the parts of prokaryotic cells and bacteria in particular, and some distinctive and unique characteristics found in those bacteria. So let's get started with microscopes. So microscopy, a microscope is simply just an instrument that we use to make things much bigger so they're easier to see. But in the world of microscopy, and there are people who are microscopy experts, that's what they do for a living. You can even get a degree in microscopy, graduate degrees. Anyway, in microscopy, we use a microscope to see things that we can't normally see with the naked eye. But in using microscopy and selecting a microscope, there is some terminology that's very important. First, what is the resolution of a microscope? And resolution is really talking about, frankly, how good is the scope, right? How much quality does this microscope have? Because the smaller the resolution, the lower the resolution number, the greater resolving power the microscope has. And what resolution really is doing is it allows you to see the difference between two different points. For example, I'm just going to use my pen here. If I have two dots that are let's say 2 micrometers apart, right? Let's just say these two dots, these are two bacteria cells that are 2 micrometers apart. With microscope A, with a resolving power of 0.1 micrometers, I can see there is space in between these two cells, and I can determine that they are two separate cells. But microscope B has a resolving power of, say, 0.5 micrometers. That would mean that these two cells would look like one large blob, that I would not be able to see that small little 2 micrometer distance between those two cells. So the resolving power for microscope B, although it's a higher number, 0.5 is greater than 0.1, with this higher number here, I can't see the space in between here because that space is less than 0.5 micrometers. So the higher the number, the less you can see, the less, not detail, but the less ability you have to distinguish 2 points from each other. Focus is oftentimes interchangeably used with the term resolution, and that is simply just how sharp, what's the quality of the image that I'm seeing. You can think of focus with a camera. If we focus with a camera lens, we want to get it nice and crisp and clear. We want to see every single little detail in the photograph. So focus is really about the sharpness or determination of detail. The ability to focus with a microscope is highly dependent on the resolving power or resolution the microscope itself is capable of. So resolution and focus are the two key components if you're ever purchasing a microscope, say for your, if you're working in a doctor's office or something and you're the office manager or in charge of the lab section. You may be asked to look into microscopes for purchase, and depending on what you're looking for, you're going to want to really concentrate on resolution and focus. Parafocal is a characteristic that most microscopes have. Ours and our labs do. You probably used the microscopes in maybe anatomy. In parafocal, this is just the ability of the microscope to remain or maintain some semblance of focus when you move from a low magnification to a higher magnification. This allows us to only have to use a fine focus knob in the microscope to be able to focus. And then finally, we have the concept of total magnification. Total magnification is used when we have what's called a binocular microscope. And a binocular microscope, actually it's a binocular compound microscope, excuse me, that's important. And total magnification is because we compound the magnification of the specimen we are observing. For example, in this picture at the top here, this is a 100x objective. We know that because it's being used with oil. So oil immersion objectives are 50x or higher. I just happen to know the one with the black ring is 100x. So this is the objective. And the objective of the microscope is kind of the muscle. So this is where we are able to adjust our magnification. We can go from 10x to 40x to 100x. So we have adjustable magnification with the objective. Below here, the picture below, this is what we call the ocular. And this is the eyepiece that we look through. And the eyepiece also magnifies our image another 10 times. So when we have a compound microscope, this one is called binocular because it has two oculars, or two eyepieces, right? We have two eyes, so we use two eyepieces. These two eyepieces are the binocular part. Compound is where we magnify the image 100 times, but when we're looking through this objective here, we're magnifying it another 10. So our total magnification is the complete compounding of what we are looking at in the specimen, which will be the magnification of our objective, 100, times the magnification of our ocular, which is 10, which gives us a total magnification of 1000x. If I were doing this with a 40x objective, I would then multiply my ocular times my 40x objective, and that would give me a magnification of 400x. Notice the objective is where we have variability in the magnification. The eyepieces always remain the same. The oculars are the same. Most microscopes are 10x, so it's pretty safe to multiply by 10, but there are a few clinical microscopes out there that are 12x to try and the idea there is to try and magnify as much as possible, because objectives, once you get past about 150x, these objectives are no longer good in light microscopy. We can't magnify much higher than that. So they'll use the objectives to try and increase and tweak that magnification just a little bit more. Now, other types, what are the different types of microscopy? Well, the one that we're all used to is the Brightfield microscope. Right here on the top left here, we have a picture of a binocular compound light microscope. This is the microscope that we use in our microbiology lab. Again, that compounding is why we magnify the image twice. So remember, this will give you your total magnification, what we refer to as TM. The standard Brightfield microscopes these days use a standard halogen light because the halogen bulbs have the highest energy wavelength of light and concentrate blue light, which is our high energy light. Of course, two oculars, two eyepieces. The maximum magnification you can get, total magnification, is around 1500x. We can use a 10x objective, and they do have, a 10x ocular. There are a few 15x oculars, and so a 15x ocular with a 100x objective would give you a complete or total magnification of 1500x. Now, the way that Brightfield microscopes work is they bounce light off of the actual specimen itself. So here in the bottom left, we have some pictures. So this is what some bacteria look like at 4x. Then we move up to 10x, then 40. So this is a total magnification of 40x. This is a total magnification of 100x. Our 40x objective would be 400x. And then here, our 100x objective would be a total magnification of 1000x. And you can see there's a really big difference. Every time we move from 40 to 100x total magnification, it doesn't look all that much different. From 100x to 400x, that's a little bit, there's definitely some significant difference here. But when we go from 400 to 1000, that is 600 times more visibility. So that's 600 times the magnification. So that's pretty impressive. And we can get down to the individual cell level at a 1000x objective. Another form of microscopy that's used quite a bit in microbiology as well as in the healthcare field is Darkfield microscopes. Darkfield microscopes usually are just Brightfield microscopes with a special type of filter on them. It has the same magnification range as a light microscope. However, we use Darkfield microscopy for specimens that are really hard to stain. Syphilis is the example we use here. Syphilis does not stain. It will not retain stain very well. And they'll use, if syphilis is suspected, they will actually use Darkfield microscopy. It is also used when we want to observe a specimen that is living, because with Darkfield microscopy, we do not have to stain the specimen. I just want to go back real quick. Notice our bacteria here are red. And that's because in nature, in vivo, bacteria do not have any color. They're colorless. So we wouldn't be able to see them. But we add stain to the bacterial cells to be able to see them under the microscope to add contrast. Well, some organisms, if they don't hold on to the stain, we're not going to be able to do that. So in dark field microscopy, it's called dark field because the background remains dark. And you can see here the specimen, light shines through the specimen as opposed to bouncing off of it. And in doing so, our specimen lights up with the background remaining dark. So it's kind of like a negative type of like the negative of a photograph. So when you have an organism that's really hard to stain, instead of trying to go through all kinds of crazy ways to stain it, just switch from bright field microscopy to dark field. Also, this can be used for specimens when we want to observe them in a living state. Staining bacteria kills it. It also can alter the shape of it a little bit. These guys are so small, it's like adding 200 coats of paint on the outside of those cells, and it can sometimes alter their shape. So if we want to really get good detail and we really want to maybe see an organism for motility, see if it's moving, has flagella, those sorts of things, we would use dark field microscopy to observe living things. If you go to the dentist, many dentists nowadays, they like to show people all of the bacteria on their teeth to try and drive home the concept of plaque and bacteria on teeth. So will take a tooth scraping, they'll kind of scrape the teeth a little bit, smear it onto a slide, and then show it to you using dark field microscopy so that you can see the bacteria. And that the beauty of that is whether the bacteria living or dead is not the point. The point is that you can actually see the microorganisms and they didn't have to go through any kind of staining process. They could just put it directly onto a dark field microscope and look right at it right then and there. Another type of microscopy that is a modified form of bright field is the phase contrast. Now, in the phase contrast microscope, this is another specialized type of filter that's added to certain bright field microscopes, and it amplifies what's known as the refractive index. So in bright field, we just let light bounce off of our specimen. We didn't really get specific with it. With dark field microscopy, we let light shine through our specimen. And again, we didn't really get very specific. But with phase contrast, we want to get more detail. So the filter that's used actually amplifies differences in refractive index or in the density of the actual specimen itself. So here at the bottom, we have a phase contrast of some algae, and you can see there's some differences here, and the background is kind of bluish gray, which we get with phase contrast, or it can look, even here, it can look like black and white, like almost close to dark field. And we can see that where the cells are more dense, like right in here, where all of these chloroplasts are inside of this algae cell, it's really dark. We get much darker color. But up here, where we have what's known as a central vacuole, up in here, this guy, we can see there's very little in there because central vacuoles store water and nutrients. So there's not as much, there's no chloroplasts in here, there's no chlorophyll or anything like that. It's less dense, so the light can reflect off of it differently. Up here in the top picture, we can see where the bacteria are. The bacteria cell itself is very, very dense, so it's very dark. And then we have these flagella. This is the beauty of a phase contrast. This is a great picture because you can see these flagella here. And flagella under dark field microscopy and bright field microscopy are very difficult to see. You have to use specialized staining processes in order to see those flagella, where in phase contrast, because we're enhancing the refractive index because of the density of something, we can actually get more detail. So we get much greater detail and resolution or resolving power than we do with phase contrast than we do with bright field or dark field. Finally, we have what's called differential interference microscopy. In DIC, we now are looking at, this is kind of like images stacked on top of each other. It's using software with a microscope, and so it's usually observed through a computer. And with DIC, you actually take a bunch of pictures, slices of the organism at different focal planes, and then stack them on top of each other. In doing that, this provides you with a more three-dimensional type of picture of the organism. So to compare, I have at the top, letter A here, the top images were made using DIC, right? So these are all DIC, and down here at the bottom is phase contrast. And I like this image because it really kind of gives you an idea of when you should use one or the other. Just because something gives you a three-dimensional effect or might provide more detail on something doesn't necessarily always mean that that's the microscope that you want to use. So for this, on the letter A here and letter B, A and B are red blood cells. Now, do I want to be able to see through the red blood cell? If I use the DIC, which is just kind of uses two different sources of light and brings them together later, I don't really see a whole lot. There's not a whole lot inside of a red blood cell, but maybe I want to see the surface of the red blood cell because I may be looking for something. In that case, I would want to use a phase contrast. I can get much greater detail here with the phase contrast than what I'm seeing with the DIC. But over here, I have some epithelial cells. In C and D, these are epithelial cells, and I want to see inside of it. So here, I'm going to select the DIC method because I get a lot more detail in here. I can see nuclei, I can see structures. These are probably small ribosomes of some kind or proteins. I can see detail in this cell that I cannot see here in the phase contrast. And finally, over here, we have a specialized type of bacteria called the cyanobacteria. And inside of here, I can see some different structures inside here, whereas on the phase contrast, I cannot. So if I'm looking for internal structures, I would want to see my DIC. But if I'm looking for external structures, then phase contrast would be fine. Now comes my absolute favorite form of microscopy, and that is fluorescent microscopy. This stuff is super cool. So in fluorescent microscopy, we can do this one of two different ways. Now there are some microorganisms that actually produce their own fluorescence, and these are microorganisms, they're bacteria. For example, if you've ever seen a cuttlefish or certain octopus, when they glow at night, they will sometimes glow. That glow is created by a chemical signal the animal sends to bacteria that produce a natural form of fluorescence. And those bacteria have a symbiotic relationship with that particular animal, and the animal sends them a chemical signal, causing the bacteria to release their fluorescence for a brief period of time. And so cuttlefish will do this to attract prey, and octopus will do that to attract prey as well. So those are forms of natural fluorescence. But somebody got the really cool idea one time. I need to look up the history of this because I don't know it. But somebody came up with the great idea one time of using the idea of fluorescence in specificity. So they take fluorescent dyes and they attach the dyes to antibodies. Now, antibodies are small proteins produced by our immune system that when they attach to something, they're very, very specific to what they attach to. If I create an antibody, if my body produces an antibody against, let's just say the flu, then those antibodies that are flu antibodies will only attach to the flu virus. So that if I were to attach a dye to the actual antibody itself, then when the antibody attached to the flu virus, the dye would be attached to that flu virus as well, but only to the flu virus. So wherever the flu virus was in my specimen would glow. Where my flu virus did not exist would not glow, unless I provided an antibody for that. So over here on the right, these are some, up here is actually tularemia. Tularemia is an intracellular obligate bacteria, and it causes the disease tularemia. And there was somebody studying this particular microorganism, and so they used fluorescent microscopy to identify tularemia bacteria inside of macrophages of mouse cells. So the bright green are antibodies tagged with a green fluorescent that are specific for tularemia bacteria, whereas the red coloring that you're seeing are antibodies with a red fluorescent dye that were specific for microskeletal elements inside of the macrophages, so that they could differentiate and literally locate where these tularemia bacteria are inside of the actual macrophages themselves. Down here at the bottom are just simply some tularemia that have been stained with the green fluorescent dye. This is not an antibody picture down here. So by tagging with fluorescent dyes on antibodies, we can get very, very specific, and we can we can stain different cell parts with different colors. This is not unique just to microbiology. Many of you might remember your mitosis pictures in the textbook or in class, and you saw these bright blue and yellow, these fluorescent-like colors. That was done using fluorescent antibody tagging. And one of the first proteins or dyes used for this is GFP. Now, GFP is green fluorescent protein, and that's actually the gene from some microorganisms that have natural fluorescence. We can now take that gene and we can introduce it into other cells. Say down here, that's what they did here. So you can introduce this gene into a bacterial cell. This would be done for research. I introduce the gene into the bacterial cell, and now that bacterial cell will produce the green fluorescent protein. I can then take these GFP producing cells and introduce them into mouse macrophages. The mouse macrophages, when the bacteria infect these macrophages, they're going to continue producing this fluorescent protein, and now I don't have to stain them. I can just look for them under the microscope. They should be glowing for me. In electron microscopy, these are the last two forms of microscopy that we're going to look at. And there are two different types of electron microscopy. I can use scanning electron microscopy, where I would look at the outside. So this down here is what we call SEM, or scanning electron microscopy. In scanning electron microscopy, I am looking at the exterior, the outside of a microorganism. With transmission electron microscopy, which is what we're looking at up here, right? So this is TEM. With TEM, I'm looking at the interior of the bacterial cell, all right? So the inside. And both of these are using electrons, not light. So there is no light in this process. And electron microscopes are kind of like big boxes like this, and they have a huge, giant, long tunnel, and they have a bunch of cameras in here. There's a camera inside of here, and then all of this is linked over here to a computer. Just draw my little computer there. So it's all linked to a computer, and these are photographs taken by the picture. Now, what's happening here is the bacteria are prepared on a slide, and instead of adding a dye or a stain for, let's take SEM first, the bacteria are adhered to a slide, and then they are dipped in a gold solution. And the heavy metal is incredibly thin, just a few nanometers thickness, in order to coat the specimen. And then the specimen is placed here into the electron microscope, and electrons are shot down at high power onto the specimen. And when they hit the specimen, depending on three-dimensional, you can see these are very three-dimensional, these electrons are going to bounce off those gold-plated specimen, and they're going to hit different sensor plates found inside of the electron microscope chamber here, and they're going to hit at different speeds and different angles. And the computer software is going to calculate all of that and use algorithms to form these types of three-dimensional pictures that you see here. So that's how an SEM works with electrons. Now, a transmission electron microscope works exactly the same way. Exactly. However, the specimen preparation is a little bit different. So in the specimen preparation here, before the bacteria are placed onto a slide and dipped in a heavy metal solution, they are sliced incredibly thin, about a micrometer, or even just a few nanometers in thickness. These thin slices are then placed onto a slide, adhered to a slide, applied an even thinner coat, maybe 1 nanometer thick gold or silver solution, some kind of heavy metal solution, and then exposed to electrons. And in addition to electrons bouncing like they do in the SEM, they also look at electrons that pass through the specimen. And that's how they get these different densities in color. Okay, so this is a transmission. So if you want to see the inside of a bacterial cell, you need to use TEM, or transmission electron microscopy. If you want to see the outside detail, maybe cilia, flagella, proteins, structure, shape, those sorts of things, then you would use scanning or SEM microscopy. Now, in order to see all of these microorganisms, we have to prepare them for microscopy themselves. And so bacteria are stained with color, or in the case of electron microscopy, they're being stained with heavy metals, but they're being stained in order to increase the contrast so we can actually see these microorganisms. They're clear. Stains are salts, so they have a charge, and bacterial cells are negatively charged. So bacteria have cell walls. The cell walls have a very strong negative charge. Some bacteria have a stronger charge than others. So if we want to actually stain the cell itself, we want to paint the cell so we can see it in color, then we use the type of dye known as a basic stain or basic dye. And basic dyes have a positive charge, so they're attracted to the negative charge of the cell walls of the bacteria, causing them to adhere to each other. If I want to look at the background, but not necessarily stain the cell itself. Remember, these guys are really tiny. So if I start adding layers of stain to the bacterial cell, I can very quickly and very easily actually alter the outside of it with that stain. So there are occasions where I can use a stain to mimic what we might see in Darkfield microscopy. That's known as a negative stain, and that type of staining method uses a stain that we call acidic or acidic dyes. With acidic dyes, the bacteria are going to repel the stain, and we don't rinse it, so that the background of the slide is nice and dark, and then the cells are not stained at all, and light will shine through the cells. So negative staining and using acidic dyes will mimic Darkfield microscopy. Staining techniques are used to enhance or to differentiate different types of bacteria from each other. In the next lecture, we're going to talk about gram staining extensively and the differences in gram staining. But in order to prepare for that, we're going to just talk about some of the different staining techniques that are used. So in staining techniques, we have three basic groups of stains. We have a simple stain that we just throw some color onto the bacteria, throw it under a microscope, and take a look at it. The problem with simple staining is very quick and easy, and it works great if you just want to see bacteria. But one of the problems with it is it's very limited in what you can determine. So the only three things you can determine using simple staining techniques is you can determine how big the cell is. You can also see what we call its arrangement, how the cells interact with each other. Do they stick to each other, or are they planktonic, meaning singular? Or the morphology, what shape are they? We have some basic shapes found in bacteria, and the actual morphology is an important part of identifying bacteria. So simple staining, we just simply use one color, throw some stain on there, and take a look and see what we have. Now with a differential stain, we're going to use two or more different colors, because one type of cell is going to stain one color, a different cell will stain a different color, and we're going to stain them based on some kind of characteristic. That's going to allow us to tell the difference between them. Now there is a type of staining in microbiology that is the gold standard in micro, and it is a differential staining technique known as gram staining. And gram staining is the very first thing that any microbiologist or laboratory technician will do in order to identify a microorganism. Gram staining is so important that when you get hospital results back from the laboratory on a microorganism causing infection, you are going to receive the gram stain result before you even get the name of the organism. It'll say gram-positive Staphylococcus. And this is important because gram staining determines the course of treatment. Gram staining is based on differences in cell wall composition. Gram-positive organisms have very thick cell walls. Gram-negative organisms have very thin cell walls. And how these organisms interact, how antibiotics affect them, is different between gram-positives and gram-negatives. So there are some antibiotics out there that are very effective against one type of gram-positive organisms, but not effective against gram-negatives. So knowing the gram stain result of a microorganism in identification is key to determining the course of treatment required for a patient with a bacterial infection. Last but certainly not least is the structural stain. We use a structural stain to look for specific structures, such as a flagella or endospore or capsule, those sorts of things. Now, we don't 100% classify a staining technique into one of these three categories. You can have something like an endospore stain that is a combination of both differential and structural. But these staining techniques are just a basic guide for the types of techniques that are used to observe bacteria. So this is the gram stain here, the gram staining procedure. I'm not going to go through it in great detail because in our next lecture in cell morphology we will, but I just want to show you the differential aspect of it. So in gram staining we have two different types of bacteria that are here together on the same slide. If a cell is gram positive, I'll find one here, then it is going to be purple. If a cell is gram negative, then it's going to be red. And the reason for this is we apply what we call our primary stain first, and that causes all the cells to be purple. We will apply some kind of mordant that will cause the purple dye, the crystal violet, to adhere to one cell type but not the other. We then go through our differential step, and that's this one right here. Just going to put that there. And decolorization is where we remove the stain from one cell type, but because of that mordant, it will be retained by the other. So you can see here, these rods, we lost the purple dye, but the cocci, the purple dye remained behind. We then add a counter stain. This counter stain is simply added so we can see the other organism. So our cocci are still purple, and we always use a lighter stain, a lighter colored stain as a counter stain. So our cocci are really purple, and now those rods that had gone clear after decolorization are now this reddish pink here that we can now observe them. So in gram staining, gram positive cells are going to stay purple, and the gram negative cells are going to be red. So just these are some questions I want you to ask yourself and a way to review. So make sure that you know the names of the 5 different types of microscopy that we studied. You should know the difference between a TEM and SEM in electron microscopy. You need to figure out how to use the different types of microscopy. So for example, if you needed to observe a living specimen, what kind of microscopy would you choose? What are fluorochromes? And why are they added to something in order to differentiate different structures in a cell? If you wanted to see an intracellular obligate pathogen, such as a virus or a bacteria, what kind of microscope would you use? Make sure you know the order of the gram staining procedure. We'll go over that several more times. What do we use gram staining to differentiate between gram positives and gram negatives? So what's the difference between a gram positive and gram negative? How do you calculate total magnification in microscopy? And if you were provided a picture or a photograph of a microscopy specimen, you should have a general idea of the type of microscopy that was used to obtain that picture. So those are just some things that you might see on an exam or on a quiz, and these are questions that you should be reviewing for the microscope section of this lecture. I hope you enjoyed the lecture, and I'll see you in the next one on cell structure.